Okay, here we are. We're going to follow lipid digestion from mouth to absorption to the body. So just to point out something that's slightly different on this video, we've got three different kinds of lipids represented. So we've got some triglycerides here, and then this little thing is representing a cholesterol molecule, which is another type of lipid that you might consume. And then these tiny little dots are representing fat-soluble vitamins. So vitamins D, E, A, and K are all fat-soluble, and they're going to be considered here in this lipid digestion. So we're going to take these three different kinds of lipids, and we're going to pop them in the mouth, and we're going to once again identify some mechanical breakdown that's going to be happening here. And so, of course, we've got the mastication, actions of the tongue, and the watery saliva. Now, something new is going to be going on here with lipid digestion as far as what's happening in the mouth, in that we are going to add a molecule here in the mouth that you could consider as being chemical digestion, except that it's not active until it hits a pH of 2. So I'm going to put it here in parentheses. We add lingual lipase, but we're going to note that we're going to note it again when we get to the stomach, because that's actually where that molecule is going to be employed. All right, so let's grab those lipids in the mouth, esophagus, peristalsis, to move us down to the stomach, where we're going to get some new actions. So, of course, one of our new actions, or one of our mechanical breakdown actions here in the stomach, is chopping with that oblique layer of the muscularis externa. And then we are going to activate lingual lipase because pH of 2. So I'm going to just write that in here. pH of 2 really gets us the action of lingual lipase. And we're also going to add a second lipid digesting enzyme here called gastric lipase. That gastric lipase was added to the lumen of the stomach from a group of cells called chief cells, and they're in the gastric glands of the stomach. All right, so the action of lingual lipase and gastric lipase together will be able to break down about 30% of the bonds. So let's see if I can get my eraser going here. So we'll break a couple bonds here. And this thing I've got here now, if you take a look at this, what used to be a triglyceride here is now I've got two free fatty acid tails, and I've got one monoglyceride. And that's really going to be the goal of breaking down these triglycerides, is to get that monoglyceride and two free fatty acid tails. We're not going to need to do any chemical breakdown of these fat-soluble vitamins, nor these cholesterol molecules. They're already small enough to absorb, so we just need to move them eventually to where they're going to be absorbed, but we don't need any chemical breakdown on either of these kinds of lipid molecules, just the triglycerides. Okay, so now let's grab our whole group of lipids, and we're going to move them forward into the small intestine. And things are going to get pretty busy here, so I'm going to go ahead and move these guys into our zoomed in view of the small intestine so that we can deal with what's going on here. Okay, so now we're in the small intestine. I want to identify both some more mechanical breakdown that's going to happen here as well as chemical digestion. This part's going to be a little bit interesting. We consider a component of the small intestine a mechanical breakdown because we need to be able to emulsify these fats that we've moved into the small intestine, and we need to separate them from each other. And so we're going to add some bile salts, and those are going to surround these lipids and sort of get them into really small packages. So I'm going to draw some bile salts in here. And as I draw these in, I want you to notice that I'm drawing sort of two different ends to the bile salts. So we have this little round end here, and in my drawing, that round end is considered hydrophilic. It is happy to be touching water. And then the little stick end that I just drew there, that end is hydrophobic, and that end is going to point toward my also hydrophobic lipids that I've got here. And we'll surround small little packages of these lipids in this manner in order to get them separated from other lipids that have been in that chyme that moved into the small intestine. Because of that, we can also identify an enzyme that's going to be able to now reach these small packages, and that enzyme is called pancreatic lipase. And that was part of the pancreatic juice secretions. And that pancreatic lipase, I'm going to just draw some tiny little circles in here. That enzyme gets packaged in with these bile salts and can start digesting the rest of the triglyceride bonds there. So I'm going to go ahead and just break the rest of my triglycerides down into monoglycerides and free fatty acid tails. This package here that we've got, I should identify a name to it. So this little package is called a Micelle. So the Micelle is the bile salts surrounding the dietary lipids and the addition of pancreatic lipase. Okay, so I'm going to grab my cholesterol and my fat-soluble vitamins and my monoglycerides and free fatty acids, and I'm going to pop them into this structure called a lacteal. And I want to explain what's happening here because you'll notice that I've left my bile salts behind. So the bile salts that make up the outside wrapping of the micelle are going to crash against this brush border and dump all of their contents into the lacteal, but the bile salts stay in the lumen of the small intestine. Those bile salts will be reabsorbed in the very last portion of the small intestine, the ileum. They'll be returned via the bloodstream to the liver, so the liver can put them back in the bile once again to be recycled. These guys right here are going to be put into this lacteal. The lacteal is a lymphatic capillary. And the way they're going to get put into that lymphatic capillary is they're going to need to be repackaged before they can get in there. And so I'm going to just grab a different pen color here. And we'll get ourselves a new little packaging. So in the cells that are lining the small intestine, those are simple columnar epithelial cells. Those simple columnar cells will pick up all of these products from the micelle, leaving, of course, the bile salts behind. And we're going to surround inside this simple columnar cell, we're going to re-surround our dietary lipids in a new package of phospholipids. And this new package that's being made by the simple columnar cell is called a chylomicron. And so now that entire package will enter this lacteal and travel via the lymph to the bloodstream. So here they go, moving through the lymphatic system, and they'll get dumped into the bloodstream with all the lymph as it moves into the bloodstream, and that bloodstream will bring that chylomicron to the liver. Once again, we're going to repackage. The liver is going to remove the outside of this chylomicron and repackage it into a new, we're going to just get rid of this. Repackage it once again, and this new package is going to be called a very low density lipoprotein. The very low density lipoprotein can now move into the bloodstream again and deliver all of these lipids to the body. So this very low density lipoprotein is going to cart around all of these products, passing them off to the cells that need them. As it passes off its contents and the contents begin to disappear, it switches to a low density lipoprotein, LDL, and eventually, once that package is empty, it will return to the liver, and the liver will pick it back up again. So if low density lipoprotein levels in the bloodstream are low, it means that this process of this package here and the delivery of that material has gone pretty efficiently, and the cells are picking up what they need and returning the empty LDLs back to the liver. Okay, one final comment to make. There's one more thing that might happen here that really seems appropriate to discuss now, and that is a different kind of package that the liver sends out to go gather lipids from the body, like cholesterol and free fatty acid tails. And this package that the liver builds to send out and gather lipids so that it can use them for things that it needs to do is called an HDL, a high-density lipoprotein. So this package goes out to the cells of the body, gathers lipids, and especially lipids like cholesterol. So I'm going to draw a little cholesterol molecule in here, just so that we can sort of imagine that that's what this thing would be gathering. And maybe some fatty acid tails in there too. Okay. The reason that the liver might gather that stuff up is because it needs to make things with this. One of the things it might need to make with it are bile acids, because bile acids are made out of cholesterol. Now, why would the liver need to make new bile acids instead of just using the recycled ones from the last meal? If some of those recycled bile acids actually were not resorbed in the ileum but were lost in the feces, then the liver is going to need to make new bile acids, and it'll go out, gather cholesterol, and build those new bile acids and put them into the gallbladder. The thing that might reduce reabsorption of these bile acids in the ileum is fiber in the diet. So fiber in the diet, such as eating oats or Cheerios, is going to trap some of these bile acids. They'll be lost in the feces, and then my liver's going to have to go send out some HDLs to get some new ones and make some new ones, which in theory would reduce the overall cholesterol in the body because we have to keep using these to make new bile salts. All right, that's the digestion, absorption, and use of lipids in the body.